Good afternoon, dear students. So today I will explain the third question of third seminar, talking about visual system, the optical system of the eye, blind spot, the pupil and pupillary reflex, accommodation, and refractive abnormalities of the eye. First, let's revise the anatomy of the eye, starting from outer structure to inner. So outer structure we have cornea and sclera. Cornea is a transparent structure which allows the light to pass. And next we have anterior chamber filled with aqueous humoral fluid. And then we have the pupil surrounded by ciliary muscle. And then we have the lens, and this lens is getting attached by suspensory ligament, and this suspensory ligament is attached to ciliary muscle. Then we have vitreous chamber, and in the end, in the back here, we have retina, which contains rods and cones, which considered our sensory cells for vision. So let's revise simple physics about how lenses work. So you know that the lens, convex lens play a role in focusing the light. So imagine that's an object, the light's coming, so they focus the image, unlike the concave, which distribute the light waves. So that's, if you remember from physics, we have refractive, what we call the refractive index, which how much the medium is thicker than the air, because you know that higher refractive index is mean light waves will get refracted to inside, to closer to each other, not away. So that's an object. The light is going through an air with a refractive index 1, and then going through cornea with the refractive index 1.38, aqueous humor, and then to the lens with refractive index 1.4. You don't need to remember these numbers, just remember that they have higher refractive index than air. And lastly, they present the image on the retina. Of course, you know that the convex lens, they produce an image upside down, and that's why this image, this information will go through optic nerve to our brain upside down until they reach the main cortex, primary cortex of vision, which is occipital lobe. And there, the image will be translated upside down, so you will get the clear image. Okay, so for vision, to see an object, we need to understand something, that we have mechanisms, different mechanisms in our eye to see the objects at different distance and different brightness. So let's start with the distance. So what is accommodation? Accommodation is our eye's ability to change the thickness, the power, refractive power of the lens depending on how far or how close is the object. Okay, what does this mean? For example, you are looking at some objects far, far away. Let's say you are going out for a walk and you saw a tree far away. So, what will happen? What will happen is we have ciliary muscle attached to or by suspensory ligaments, and this suspensory ligament attached to the lens. So we want this lens to get thicker, to have more refractive power, so you can see far objects. So what your body try to do, it will try to relax ciliary muscle, and with the relax of ciliary muscle, suspensory ligament will, so sorry for so focus with me because this part will be a little bit confusing. So let's start again about accommodation. So accommodation is controlled by parasympathetic nerves. So when you are, if you remember, parasympathetic system is rest and digest, and when you are resting, you are looking at near objects. And to look at near objects, you need to have higher refractive power with a thicker lens. That's why the parasympathetic nervous system will activate ciliary muscle, leading them to contract. So here, the ciliary muscle will contract, and with their contraction, this ligament, imagine with me, this ligament will become relaxed. Okay, and with this relaxation, the lens will not be stretched, so they will become more thick. That's for near objects, when parasympathetic nervous system is getting activated. When you are looking at near objects and when you are looking at far objects, it's vice versa. It means ciliary muscle will relax, stretching suspensory ligament, and this stretching of suspensory ligament will lead to flattening, let's say, of the lens to look at far objects. And that's the idea of accommodation, which is our ability to change the diameter of our lenses to look at near object and far object. Another reflex we have in our eye is pupillary reflex. Pupil is this gap around the iris, which it's like a gate or a window. You can imagine pupil as a window, which controls how much light is entering. So imagine today is a sunny day, a lot of light is entering your room. What you will do? Simply close the window. Exactly, that's what our eyes do. When there is a bright object or bright light, our muscles around the eye, which we have two muscles, sphincter muscle and dilator muscle. Sphincter muscle get contracted to reduce the size of the pupil, to reduce the amount of light entering the eye. If you are looking at light, like bright light, in normal, it's normal. And when you are in dark room, for example, what you will try to do? Try to let as much as light as you can to go inside. That's what the pupil do. So the dilator muscle will contract with the relaxation of sphincter muscle, leading to wider pupil, and this wide pupil will let more light inside. And that's pupil reflex. So let's revise again: accommodation and pupil reflex. Accommodation, activated by parasympathetic nervous system when you are looking at near object, which will lead to contraction of ciliary muscle, relaxation of suspensory ligament, thickening of lens to have more focus power on the retina. Okay, the other way for far object. Far object, relaxation of the ciliary muscle, stretching of the suspensory ligament, flattening of the lens. Okay, pupil, smaller in bright light, larger in dark light or dark area. And with these two mechanisms, now we can focus the image sharp and clear on the retina. So let's see the structure of the retina. So that's the direction of the light. So the light, imagine the eye is here, like for example, the rest of the structure of eye is here, and the light is entering this way. And it's upside down structure. So the rods and cones, which is the cells responsible for the light, are in the deepest layer. So the light travels through all this area to pigmented layer, and after that, reacting with the rods and cones, depending on the chemical reaction which will happen in them, some ion channel gates will open, and which will lead to generation of action potential going to bipolar cell to ganglion cell, and the axons of ganglion cells is forming what we call is optic nerve. So axons of ganglion cells forming the optic nerve. What about the other cells, horizontal cell and amacrine cell? They play a role in the vision of different colors in different uh, brightness. What does it mean? So you know that the cone are responsible for bright color, like bright light and colors, while rods are responsible for vision in dark areas. So this horizontal cell, if you're in, let's say, dark room, Rods will be activated, but some part of the cone are activated, and the horizontal cell will link these two cells, so they will give you the ability to see colors, some color, not all color, some colors in dark room. That's why, for example, in dark room you may recognize green or bright colors. It means it's because that some cones are activated and rods are activated together. And that's the general structure. For the mechanism of how this potential are generated, it will be explained in the future question. Now let's talk about refractive errors or abnormalities. So we have normal vision, where the light is focused on the normal area of the retina. But we have myopia, or what we call is nearsightedness, where the light is focused in front of the retina. And that's what most people suffer from. Hyperopia is like when the light is focused behind the retina. It's not like behind the retina literally, but like the image which is created on the retina is not clear. And that's usually you can see it in old people. And astigmatism, which when we have the light focused on not one point, but on multiple points. Sorry for interruption. So types of refractive errors. So hyperopia is the image is created or focused behind the retina. That's why the what we call is farsightedness. And astigmatism and anti or what we call other astigmatism is when we have multiple pictures on the retina, not one but multiple focus points on the retina. So the picture will appear like blurry, or like you have three things. Like if you're looking at one pen, you may see three pictures of the pen. And the idea of each refractive error is related to the same mechanism of how lens focuses. So myopia or farsightedness or is when your lens cannot get thicker to accommodate with near objects. And hyperopia, the lens uh, cannot get flattened, so you will not be able to see far objects. If you have any question about this part, you may ask me in the chat. I will provide you with some chat for questions and answers. And lastly is presbyopia. It's usually, you know it, but in other words, you usually see it in older people. That when the older the people get, the thicker and the less elastic the lens become. So imagine the lens is less elastic and more thicker. So they will have no accommodation, almost no accommodation. They will have some fixed point. So they will see object clear at some distance. Near, more near this than this distance, they will not be able to see it clear. Far than this distance, they will not be able to see it clear. And you can know about this when you see them, that older people, for example, having reading glasses to look at near objects. So they can see at medium range, but they cannot see near distance usually. And that's the mechanism of presbyopia. If you have any question about this topic, you can just, I will provide you with some chat to question and answer about the visual system. Thank you for listening. Sorry, I missed one point, basic point, but I missed it, which is blind spot. So what is the blind spot? If you remember from the structure of retina, we say that the ganglion cells axons are making the optic nerve. And this optic nerve is going through the whole retina. Okay, the axons, I mean, they are collecting until they make the optic nerve, and this optic nerve are going to the cranium and then to the CNS and so on. So the point where these axons are collected together, there is no rods and cones, only axons of the ganglion cells. So it has no retina, no rods and cones, no sensory cells, which mean we cannot sense light in this area, and that's what we call blind spot. Why it's blind spot? Because if the picture get generated on or focused on this area, you will not see the object. Your brain will try to fill the object, like this gap in the vision, with some just background, but you will not see the object, and that's what we call blind spot. And with the practical classes, if I'm not mistaken, you will have a class to measure how large is your blind spot and its location. And thank you for listening.